Now in our course we turn to these final two concepts, and we'll talk about these and wrap these back to the other themes in our course, but we're already sort of pointing toward the end of our course. Lots of more material to go to get there, but the key thing is that we're going to start talking about DNA, and DNA is the primary heritable molecule in all cells. So all life is composed of cells. All cells come from pre-existing cells. We've just learned a lot about that. Those cells exist in a water environment because all life on our planet shares a very similar chemistry. A good feature of that is how the cells are all bounded by a cell membrane composed of a phospholipid bilayer. You know, lots of commonality in all life. And then that chemistry of life features lots and lots of talk about proteins. And so it's not surprising when we turn to DNA as the primary heritable molecule and we ask, what are we inheriting from our parents? And primarily, we're inheriting the code to code for human proteins. Human insulin, human ATP synthase, human LDL receptor, and so forth. So we're going to learn about that in chapter 17. And then we're going to learn about the concept of gene regulation in chapter 18, and a concept we'll talk about called totipotency. So these are featured really in chapter 17 and 18, and here's the order that we're going to go through these chapters. Chapter 17 is about protein synthesis, so zooming in on the ribosome and learning about how the ribosome uses the mRNA template to code for a protein. Then chapter 18 will explain gene regulation. Insulin is only made in the pancreas. Oh, let's just deal with this right now. Insulin is only made in the pancreas, but each of our cells has the insulin gene. How is that possible? The answer is gene regulation. Not only is insulin only made in the pancreas, but it's only made at a certain time in our life. It's made most of our life that we think about, but for example, embryos aren't making insulin. So at some point in your development as a fetus, you activated the insulin gene. All your cells had it up to then, but it wasn't being made. Now the insulin protein is being made. All the cells have it, but it's only made in the pancreas. So we get very interested in gene regulation, and then the concept totipotency is that all cells have the insulin gene. All cells have sort of this total potency, in theory, the total potential to make all the different proteins in humans, but they're only making a subset of them. So how does that work? How does totipotency work? And the answer is totipotency works by gene regulation. The cells only are using a subset of that DNA. So we're really interested in that. Chapter 17, chapter 18. And then that covers what scientists refer to as molecular genetics. And in chapters 14, let's see. Yeah, then in chapters 14 and 15, we'll learn more about traditional genetics that you would have had some exposure to already. Like, you cross a red flower and a white flower and you get a pink flower. Traditional genetics is sometimes called Mendelian, after the origin, Mendelian genetics, or sometimes called transmission genetics. And it's all about doing crosses and crossbreeding and seeing what the results are as a result of a cross. So we start out next with molecular genetics. We're ready to deal with that because of all our focus on molecules and proteins. Then we'll go back and go backwards into the text, and it's actually easier to understand Mendelian genetics once you've gotten exposure to molecular genetics. So we'll get that taken care of. Then Chapter 16 is about DNA replication. So it's really a different chapter. And scientists work with DNA and manipulate a lot. So that chapter sets us up to go to chapter 20, which is about sort of biotechnology, which is often involves manipulating DNA. And you know, DNA is physical, or I should say that DNA is physical. We'll stress that over and over again. And you know, once we get a hold of it, we can manipulate it and alter it, or mutations can occur and cause trouble. Okay, so that's our flow, and you go back to this woman, and we go into chapter 17, 18 with this interest. And this interest is that she's got cancer because there was a DNA of one chromosome and DNA of another chromosome, and those DNAs accidentally got broken and rejoined, and something called a translocation. A piece of DNA was translocated, and so now she's got some DNA from one chromosome joined to some DNA from another chromosome, and you can see that under a microscope, and you can also zoom in and study the letters, the AGCT code, and you can zoom in on the details, like which page in the book was torn and glued together with another page. And scientists know a lot about that. In her case, they know that there was one part of a collagen gene was joined together with the PDGF gene. And we can talk right now about what's going on here. The PDGF gene portion, well, let's just talk about this idea that all genes, PDGF gene, collagen gene, is an important concept. All genes are made of DNA. So we've drawn some DNA, stranded DNA, and it's double-stranded DNA. So that twisted helical ribbon of DNA, and it's composed of A, G, CT letters, nucleotide letters in a pattern, and maybe AAA, C, TA. Patterns that organize this information. And all genes are made of DNA and are composed of two parts. One part is called the transcription unit. And that's the business part of a gene. That's the part that gets transcribed into, gets copied into RNA. And for a protein coding gene, that RNA gets copied into a protein. So that's why I say it's the business gene, part of the gene. For PDGF, this is the region that has the information to code for the protein. That region of the transcription unit, subset of the transcription unit, is called, the region that codes for a protein is called the coding region. And that's dealt with in chapter 17. The second part of a gene, every gene has, is generally called the promoter. And that's the regulator. So when we say, hey, we're interested in insulin, we're interested in two things about it. We're interested in the protein that gets made, and we're interested in the idea that it was only made in the pancreas and only made at certain times and certain levels. And so what's happened in this woman is that the transcription unit, the business part of PDGF, has gotten joined to the promoter for collagen. Normally, PDGF is made in the bone marrow. In megakaryocytes, we learned about that. Normally, this collagen gene is made in skin. The skin cells have the PDGF gene, but it's not made there because it's not active there. The skin collagen gene is active there, and what happens in this woman with cancer, in this form of skin cancer, the promoter of skin collagen gets fused with the transcription unit of PDGF, and now skin cells make PDGF. That's her problem, is that because this DNA promoter activates whatever is next to it, and now the PDGF gene is next to it, she is going to make PDGF in the skin and then release it through exocytosis, and that PDGF will bind to the PDGF receptor and stimulate the cell cycle. So we get interested in chapter 17 and 18 because that goes into the problem that that woman has. Okay, so we're going to stop there and we'll go on talking about her in DNA in the next mini lecture.